Hello everyone. In this class, we'll learn about reticular activating system. The learning objectives of this class would be to understand the functional organization of brainstem reticular formation, remember the components and functions of brainstem reticular activating system, list the functions of brainstem reticular formation, and appreciate the importance of reticular activating system in body physiology. Organization of reticular formation. In the brainstem, there are specific groups of cell bodies of neurons that form various nuclei for cranial nerves, that is cranial nerve nuclei, integration for motor and sensory activities, that is brainstem motor nuclei, Oliver nucleus, etc., and relay nuclei for ascending or descending pathways. Excluding these specific cell groups, there are cell groups in the core of the brainstem that constitutes brainstem reticular formation. This reticular formation is formed by diffuse system of neurons having widely branching axons. Characteristically, these neurons have very long dendrites and axons. They have long ascending branches projecting to the thalamus, hypothalamus, cortex, and long descending branches projecting to the spinal cord. Nuclear column and their connections. The neurons are organized in two columns, medial and lateral columns. Medial column, medial group of cells which constitutes a gigantocellular part, that is large cell group that contains the raphe nuclei and central group of nuclei. They receive afferents from all sensory pathways, especially from the spinoreticular tracts. Fibers from this part project upwards as reticular activating system. They project downward as a medial and lateral reticulospinal tract. You see lateral column, it is laterally placed and is the parvocellular part containing small celled nuclei. This part contributes to sleep, awake, fullness. So here are the brainstem reticular nuclei. So these are the pontine and these are the medullary reticular formation, and this is the midbrain reticular formation. So where they are going to receive information from the different parts of the body, and they project these sensation from the thalamus by non-specific or specific pathways. These are the afferents and efferents to the reticular formation. So these are some of the afferents which are mainly through the spinal cord and collaterals from the ascending tracts. Then there will be also from collaterals from cranial nerves, including vestibular and cochlear nerves, corpus striatum, and cerebral cortex. Then thalamus, hypothalamus, and subthalamus. There will be inputs from limbic system, including septal areas, amygdaloid complex, and hippocampus, and also from the cerebellum. The main output mainly to the Oliver nucleus, to the red nucleus, and to the thalamus, hypothalamus, subthalamus, cerebellum, and autonomic centers in the brainstem, and then autonomic centers and interneurons in the spinal cord. Functions of reticular formation. They're important for control of motor activities. Reticular formation gives rise reticular spinal tract, and reticular spinal tracts are important components of medial system pathways that are involved in regulation of posture. Fibers of RST originate from the large-celled neurons of reticular formation. Motor cortex, basal ganglia, and cerebellum control posture by influencing the activity of reticulospinal tracts. There are two reticulospinal tracts, that is pontine and medullary. Pontine reticulospinal tract facilitates the spinal stretch reflexes by exciting the extensor group of muscles, that is anti-gravity muscles, and therefore important for maintaining posture. Whereas medullary reticulospinal tract inhibits spinal motor neurons that innervate extensor group of muscles. The reticulospinal tract are major components of the extrapyramidal pathways through which the brainstem controls posture. Control of sensory activities. Many of the ascending sensory pathways provide their collaterals to reticular formation. Therefore, when a sensory stimulus is applied, it is not only evokes conscious perception of the sensation but also By activating reticular activating system, it makes the individual aware of the nature and other aspects of sensation. Especially for the pain sensation, along with the alertness for the sensation, there is a simultaneous activation of endogenous analgesic system. The neurons of raphe nucleus in reticular formation are activated by paleospinothalamic tract and spinoreticulothalamic fibers. Consequently, activation of raphe spinal pathway inhibits the transmission of pain in the dorsal horn of the spinal cord. Then control of cardiovascular and respiratory centers. The cardiovascular and respiratory centers are located in the brainstem reticular formation. The vasomotor center located in the medullary reticular formation controls heart rate, cardiac output, and vascular tone. Therefore, VMC plays a critical role in the regulation of blood pressure. Cardioinhibitory center also regulate the heart functions and blood pressure. Respiratory centers, the inspiratory and expiratory group of neurons are located in the central gigantocellular cells in the medulla. The pneumotaxic and apneustic centers are located in the pontine reticular formation. All these centers control various aspects of respiration. Control of visceral functions. Visceral functions are controlled by mainly the hypothalamus. Reticular formation through its extensive hypothalamic connections controls the visceral functions. Vomiting and swallowing reflexes are integrated in the medullary reticular formation. Sleep and wakefulness. Fibers projecting from reticular formation to the cortex via nonspecific thalamic nuclei maintain wakefulness. Therefore, this neuronal system is called as reticular activating system, that is RAS. The decreased activity of RAS induces sleep and damage to the RAS causes coma. The components of reticular activating system. There are four major components of reticular activating system, that is ascending reticular activating neurons from central group of reticular nuclei, that is magnocellular part. Ascending neurons project to the interlaminar and midline nuclei of the thalamus, which is non-specific nucleus. From these non-specific thalamic nuclei, fibers project to the all parts of the cerebral cortex and limbic system. Few fibers from reticular formation bypass thalamus and project directly to the cortex. These neuronal projections form the major part of the brainstem ascending reticular activating system, which maintains arousal of the being. Increased activity in these projecting neurons causes excitation of cortical neurons and creates alertness and wakeful state. Diminished activity in these systems induces sleep. Then monoaminergic and cholinergic fibers. Locus ceruleus is located in the reticular formation at the junction between pons and medulla. Non-adrenaline secreting neurons originate from locus ceruleus. Major monoaminergic and cholinergic systems of the brain are located in the reticular activating system. These fibers are involved in integration of various sensory and behavioral activities. Fibers from parvocellular part. Fibers originating from small cell neurons located in the lateral part of the brainstem reticular formation, that is parvocellular part, are active only during the awakened state and silent during sleep. This indicates that they control sleep wakefulness. Serotonergic neurons. These serotonergic neurons originating from the raphe nuclei project to all parts of the CNS. These neurons are active during deep sleep. This indicates that these neurons are involved in genesis of sleep. Histaminergic neurons projecting from hypothalamus to all parts of the CNS play a major role in arousal. This is in brief about reticular formation. Thank you.